Good morning, everyone, and welcome to today's session. Thank you so much for taking the time to join us here this morning at this InGreen event, which is called Closing the Loop, Transforming Paper Milling Side Streams into High-Value Prebiotics and Bio-based Plastics. So we have a really nice program of events for you this morning in this session, which is going to talk about a fantastic industry collaboration from the EU-funded InGreen project. So I'm just going to introduce myself. So my name is Helena McMahon. I'm based at Munster Technological University, where I'm a head of department, and I also co-lead the Circular Bioeconomy Research Group. Munster Technological University is involved in this InGreen project. I'm one of the principal investigators, and I'm joined by my colleague, Eve Savage, who's also a key member of the InGreen team at MTU and leads out on engagement and communications. So just a little bit of background before we get to the speakers. So just the InGreen project. So the InGreen project has been running now since 2018. It's a really nice example of an industry collaboration, and all of the speakers that we've with us here today are from industry across Europe. So the InGreen project received 8 million euros in funding from the Bio-based Industries Joint Undertaking in 2018 to set up a project that would explore the potential to create new bio-based products from side streams from a number of industries located across Europe. And we hear this notion of the development of bio-based products and the development of new value chains as a really good example of the bioeconomy in action. And we can all really understand what that means with the concept really of taking high-tech technologies to convert side streams from different industries into high-value products that then can be commercialized to replace fossil based products that are on the market. And we're all well aware of the climate and environmental benefits that come from the sustainable production of these new bio-based products. So the European Commission really liked the project that was submitted. It's focused on 3 key side streams. So it looks at side streams from the dairy industry. It looks at side streams from agri-food, in particular from wheat milling. And it also looks at side streams from the paper milling industries. And it's been working away over the past 4 years to develop new technologies at demonstration scale. So this is the scale with which you are working in an industrially relevant environment to scale the technologies to see if you really can, I suppose, further scale them to large industrial scale. And this really is the kind of interesting point about today's session here, that we're going to be talking about, the scaling of these new technologies, which is hugely exciting. So it's a very large consortium. It has a mix of universities, SMEs, and multinational companies. So it's led by Narinder Barnes from Innoven, and Narinder and Innoven are based in the UK. And then we also have a scientific coordinator in the project. And we have an absolute powerhouse, Professor Rosalba Lanciati, who is coordinating this hugely complex project from a scientific perspective. And she's doing a fantastic job. And we all really enjoy working with Professor Rosalba. And I'm sure she's in the audience here with us today. So just hello to Rosalba and all of our wider InGreen colleagues that are with us here today. So then the consortium is a really nice mix of the multinationals. So you can see here we have Barilla and we have Smurfit Kappa, and then we have a range of really, really innovative SMEs across Europe that are involved in the project. And we have fantastic companies with us here today who are going to talk about their journey on the project and how they have worked with these larger companies to create really innovative new projects. So we have companies such as Avecom, NovaID, Innoven, Tecnopack, which are with us today, but we also have companies such as Mbambeli, Depot Pharma, and Paveti, who are also key parts of the project, of course, Avecom. And then we also have EFOST, who are a key dissemination and network partner in the project. So you can see really multidisciplinary and a really nice spread of industries distributed right across Europe. So I mentioned that the project was funded to work on 3 key new value chains, and it's developing a number of different products within each of those value chains. So we have the whey value chain. So this is all around looking at opportunities to deploy technologies to create new products from whey. Some of you may have attended the event that we had back in October, in which we shared with you how bio-based ingredients such as LBA, GOS, and Y lipolytica were being produced and how they were being used to generate new pharmaceutical, cosmetic, and functional cheese products. If you haven't seen that event, I'm pretty sure it's available on our YouTube channel. So I would encourage you to go and look at that if you're interested in opportunities for whey valorization. Then the second area is wheat and rye bran side streams, obviously from Barilla. The project has been developing really innovative fermentation strategies and technologies for adding value to these particular ingredients. And there is very progressed NPD underway really on taking those ingredients and converting them into nutraceutical and functional bakery products. And then we have the focus of today's event, which is looking specifically at the paper milling wastewater stream. And this is looking at opportunities to create really interesting new ingredients, PHAs, and converting those into animal feed and packaging film. And the consortium members that are here today to talk about their journey and their collaboration over the past 4 years, we have Avecom, we have Tecnopackaging, we have NovaID, Innoven, and we have Smurfit Kappa. So really nice examples of the very large multinational coming together with the innovative SMEs to create new high value products. So these are our speakers here today. So we have Jesus Orgamero, and Jesus is from Innoven. We have Luca Santorlaschi, who is from Smurfit Kappa in Italy. We have Nidia Lurenko, who's from NovaID. And these three speakers, lovely colleagues of ours from the InGreen project, are going to focus really on the PHA production and the PHA molecules and give you a nice, I suppose, insights into the opportunities and challenges around the production of this particular molecule. And then we have Irene Serrano, who's from Tecnopackaging, and Kim Windy from Avecom, and they're going to talk more around, I suppose, the product end of the story and of the value chain, and how you take those PHAs, which Jesus, Luca, and Nidia have been working on, and to convert those into the product end. So Irene will be talking around the packaging applications, and Kim will be talking around the animal feed applications of PHAs. So really looking forward to hearing all of the presentations. We're going to have a Q&A session at the end. So please pop all of your questions into the chat box, and we will take those in the Q&A session. Myself and Eve will be monitoring and moderating those questions over the course of the session. So I really now am delighted to ask my colleague Jesus to upload his slides and to get ready for his presentation, the first of the day. And Jesus is going to be speaking to you around PHAs and sharing his insights and knowledge with those as a potential replacement for conventional plastics. So I'm going to hand over to you, Jesus.
Thank you. Thank you. Okay. Can you see my screen?
That's perfect.
Okay. Okay. Can you see my presentation?
It's all good, yep.
Okay. Good morning, everyone. I'm Jesus Rodriguez. In this opportunity, I'm here to represent Innoven, where I work as a biotechnologist and research. So now I will tell you a little about Innoven, and then I will start with the real presentation. What is Innoven? Innoven is an innovation startup founded in 2013 at the University of Verona, specifically in the biotechnology department. We worked in the field of development and validation of environmental biotechnologies, with the objective to reduce management costs for the treatment of waste, with the simultaneous recovery of material and energy like biogas, volatile fatty acids, biopolymer, and other, in view of the circular economy. In this perspective, we have collaborated with a lot of European projects from our foundation until now. Between all projects, we can find Afterlife, GloPack, InGreen, usable packaging, and others. And we worked in this project specifically in the PHA research and PHA production. In addition, we produce service at the external company where we analyze their waste in order to assess their ability to produce value-added products. As you can see in the slide, through laboratory analysis and experiments, we analyzed and determined the capacity of this waste to produce, for example, biogas, volatile fatty acid, and we analyzed this experiment, and we can make the scale up of this process, and we can do experiment at a pilot plant scale. Sorry. My presentation today about the polyhydroxydocanoate production is divided in 3 topics. The first topic is the general knowledge of the PHA. The second topic is a method of PHA production. And the last topic is the currently use of the PHA. Sorry, wait a minute. Okay. The problem with the plastic synthetics. It's difficult to imagine our life without the plastic synthetics because they are present almost everywhere. Because the plastic synthetic have very spectacular properties. For example, our elasticity, resistance. They have a low coefficient of friction and other. But the plastic synthetic have a problem because the plastic synthetics are not biodegradable. This problem with the plastic synthetic causes that its accumulation in the air and causing very pollution problem in the environment. In this slide, we can see that the global production is around 400 tons per the year 2015 and increase as the years go. And the effort to control this problem with the plastic synthetic, for example, the recycle, incineration, are not effective to reduce the plastics pollution. And it's very important to say that this material are petrol oil origin, and someday the petrol oil ends in the world. So it's necessary to find the substitution of this material. The European Bioplastic Association of the 2018 concluded in order to obtain this objective and the substitution of the plastic synthetics, the material could be bio-based origin and biodegradable. So these materials are situated in this area, and in between all biopolymer from bio-based origin and biodegradable, we can find the polyhydroxydocanoate. This material is our material of interest today and in our project. So, but what are the PHAs so important? In general line, the PHA are so important because they are material 100% of biological origin, synthesized by microorganisms inside the cell when they are under stress condition. In other, we can find more than 111 microorganisms PHA producer. They are microorganisms with the capacity of accumulate 90% of its weight in PHA, and the PHA producing bacteria have been found in all environmental conditions. Concerning the property of the PHA, as mentioned about the polyhydroxydocanoates are biological origin. They are biodegradable and compostable. The polyhydroxydalkanoate can be have a stress cracking, a heat resistant, transparency, and permeability to oxygen, UV stable. They can have mechanical, physical, chemical, electrical properties similar to the polypropylene, for example. And in my opinion, the most important properties of the polyhydroxydalkanoate is the biocompatibility. We can find more than 150 different monomers of the PHA, but the three most important at the industrial level are 3 or 4 hydroxydobutyrate and 3 hydroxydovalerate. That if we mix with combine these 3 monomers, we can to obtain polymer with excellent properties, for example, elasticity, stress cracking, heavy material, depending the concentration of its monomer in the polymer. Polyhydroxydalkanoate have a problem. Unfortunately, the cost of PHA are very high, approximately 50 times higher than synthetic plastic. And this cause limiting its introduction in the market. So the 2 points that influence the polyhydroxydalkanoate cost is the high substrate cost using as carbon source for the biomass growth and PHA accumulation. And the second point that influence the cost is the PHA extraction recovery method. Usually at the industrial level, using glucose, for example, as carbon source, and the glucose is very, very cost substrate for the PHA production. These 2 points represent approximately the 18% of the production costs of the PHA production cost. So project, European project, and scientists in the world looking for solution this problem, the PHA cost problem. So, and for example, the best example is the InGreen project. We use InGreen project using low-cost substrate for the PHA production. For example, the wastewater from paper mill. And these waste have a zero cost. This low-cost substrate can to reduce the PHA production, the PHA cost. This waste is rich in BFA, also the volatile fatty acid. The volatile fatty acid are the precursor for the PHA synthesis in the microorganism. In this slide, I represent more or less the 2 methods, the typical method of PHA production. Obviously, this method can have a modification depending the industry, the application, what industry applies this production. For example, the first method is the microbial mixed culture, and the second method is the pure culture. The difference between this method is the microbial mixed culture is not a sterilized process. And the second method, and in microbial mixed culture, using different microorganisms. The difference with pure culture is in this process is under sterilization and using only one microorganism, PHA producer. More or less, I explained the function of the pure culture method. Usually, the waste is fermented in the first step with the objective to produce the volatile fatty acid. This volatile fatty acid is sterilized and then used for the biomass, for the grow biomass. In this method, we have only one microorganism PHA producer, and for the grow biomass is inoculate in the optimal condition. I mean, optimal carbon source condition, macro and micronutrients, temperature, etc., etc. After obtaining the maximum quantity of the biomass, this biomass is recovered and then inoculated in the second step. This step, the function is of this step is the polyhydroxydalkanoate synthesis. As I mentioned before about to stimulate the PHA synthesis, the microorganisms needed the stress condition in the media. So to stimulate the stress condition, we eliminate, for example, the macronutrient in the media. And in this moment, the microorganisms start to accumulate the PHA inside the cell. Then we obtain the maximum concentration of the PHA inside the cell. The biomass is hydrolyzed, the PHA is recovered and extracted. This process is under sterilization. That means is caused because the sterilization process, but you can to obtain high yield of the PHA production. In the mixed microbial culture is very similar, but without sterilization. The first step is the fermentation of the waste for BFA production. Then the BFA is rich fermented is used for the biomass selection. In this case, we have different microorganisms, and we need to select the PHA producer bacteria. So the process for the selection, the biomass selection, call it Feast-Famine, but I don't have a time to explain the Feast-Famine. But after selection, the biomass is inoculated or passed for the second step. In this step, the biomass is stimulate for the PHA synthesis, very similar at the pure culture. Then when we have the maximum concentration of PHA in the biomass is hydrolyzed and PHA is recovered. This method is not under sterilization. It's cheaper, but the yield are very low at different pure culture. So currently use of the PHA at industrial level and research tested the PHA, the PHA-rich biomass, or the pure PHA. For example, at industrial level, right now the PHA of the objective to use this PHA is for produce packaging, using pure PHA, cosmetic, antiflocculant agent, denitrified. At biomedical level, the PHA are used for biomedical implant, for example, to make implant of the ear, nose, etc., etc. Bond can be used also to make therapeutic vector for the pill. For example, I don't know if you know the pill made with the plastic. This plastic can be our polyhydroxydalkanoate. Another, the PHA-rich biomass are used for produce, for example, probiotic animal food, are used as food in aquaculture, are used also for antiflocculant agent or for the natrified, etc., etc. In this slide, I just to show you some company and a spin-off dedicated to the production of the PHA in the world. We can see that have some industry that dedicate to PHA production or dedicate to PHA research. And I concluded my presentation with the question, PHA is the future substitute for the synthetic plastic? I respond this question in the final of presentation. Thank you for your attention.
Thank you, Jesus, for that. That was absolutely fascinating. I think I had no idea personally the amount of applications that PHA molecules have. So that's one of my key takeaways from your presentation. So up next, we'll have Luca Santorlassi, and Luca is from Smurfit Kappa, and Luca is going to speak to us today around the production and scale up of PHA production. So Luca, if you'd like to upload your presentation.
Okay, can you hear me?
We can hear you perfectly, yeah.
Okay. Can you see my screen?
That's perfect, Luca, yeah, all good.
Okay. Thank you. Good morning to all of you, and thank you very much for your participation to this event. My name is Luca Santorlassi, and I'm a coordinator for the InGreen project in Smurfit Kappa, Italy. In this presentation, I'm going to give a short explanation of the industrial process for the PHA-rich biomass production that we are developing within the InGreen project. First of all, I would like to tell you something about Smurfit Kappa Group. Smurfit Kappa is one of the leading providers of paper-based packaging in the world, with 36 countries, with operations in 36 countries and with 48,000 of employees in the world. We use our leading experience to develop sustainable paper-based packaging solutions that are renewable, recyclable, and biodegradable, thus helping our customers to reduce their environmental impacts by sustaining the concept of circularity. Moreover, we continually strive to reduce the environmental impacts of our processes in terms of deforestation, carbon emissions, waste production, and water consumption. Generally speaking, the pulp and paper industry is currently considered as the third worst largest consumer of water and the sixth largest polluter of the world. In fact, a medium paper mill factory for each kilogram of recycled cardboard produces about 5 liters of paper mill wastewaters, which need several purification stages and represent environmental and economical costs for the paper production process. However, interesting opportunities can emerge from these wastes when we start to consider them as raw material for the production of value-added byproducts. The paper mill wastewater usually contains suspended solids like fibers and cellulosic materials, inorganic compounds, mainly represented by the calcium carbonate, and many organic compounds that can be referred as the COD or the chemical oxygen demand of the waste stream. In the case of Smurfit Kappa Italy, about 40% of the overall COD consists of volatile fatty acids, which represent a useful substrate for the PHA production using mixed microbial culture techniques. The objective of the InGreen project is precisely the valorization of agro-industrial wastes for the production of functional and innovative ingredients. And one of the goals of this project is to develop a sustainable process for the production of the PHA-rich biomass for different industrial applications, starting from Smurfit Kappa Italy paper mill wastewater. The process currently the process has been developed up to the pilot scale, and Smurfit Kappa Italy has a pilot unit for the PHA-rich biomass production in their paper mill factory in Pontallania in Italy. The pilot unit consists of 3 sequencing batch reactors of 6 cubic meters, and that you can see in the picture. Since the PHA producing bacteria are aerobic organisms, the pilot unit has been equipped with blowers for the air insufflation into the fermentation media. And the main process parameters like the dissolved oxygen concentration, the pH, and the volume of each reactors can be monitored by different sensors which are immersed in the fermentation media. Moreover, Two centrifugal pumps and many automated solenoid valves are used for the biomass transfer and the dosage of the paper mill wastewater into the reactors. The adapted production process is based on three main stages. First, the selection of the PHA-producing bacteria from the overall microbial population. Secondly, the PHA intracellular accumulation process. And finally, the recovery of the PHA-rich biomass from the fermentation media, so the downstream processing. This selection of the PHA-producing bacteria can be obtained by the so-called feast-famine cycles. During the feast phase, the feed is provided and all the microbial species start to consume the volatile fatty acids in the paper mill wastewater for cell growth and reproduction. When the VFA have been completely consumed, the famine phase starts and no more feed is provided to the microorganisms. In this way, for quite long famine periods, only species which can synthesize intracellular PHA as feed reserve can survive, while the majority of other undesired species will die. In particular, during the feast phase, the paper mill wastewater and the nutrient solution which contains nitrogen and phosphorus are dosed into the SPR1, so the biomass start to consume the volatile fatty acids in the paper mill wastewater to produce the intracellular PHA, and the dissolved oxygen concentration of the fermentation media decrease. When the VFA have been completely consumed, the biomass, the oxygen concentration, the dissolved oxygen concentration rise again, and the biomass enter to the famine phase. In this phase, the bacteria start to consume the volatile fatty acids, start to consume the intracellular PHA to survive. After the famine phase, part of the biomass is discharged from the SPR1 into the SPR2, where large quantities of paper mill wastewater are dosed. Since the bacteria needs high concentration of nitrogen and phosphorus in order to consume the intracellular PHA and for the PHA degradation and consumption, in this phase, no nutrients are provided, thus promoting the intracellular PHA accumulation process. When the PHA accumulation is completed, the biomass is dehydrated using a centrifuge in order to get a sludge with a solid content of about 28%. Then the residual 80% of water is removed by keeping the wet biomass at 60 Celsius degree for a few days until the complete drying on the sludge. And finally, the dry sludge is grinded using an electric mill in order to get a fine powder which can be easily processed for further steps. Here there is a small video tour of Smurfit Kappa facilities, which can allow you to better understand the entire production process for the PHA-rich biomass. Okay, my presentation is over. Thank you very much for your kind attention. If you have any question, you can contact me on this mail address, and you can also visit the InGreen project website or the Smurfit Kappa group website. Thank you again.
Thank you, Luca, for that really fantastic presentation. I think the video was so enlightening and really brought to life, I suppose, the work that the InGreen project is doing within Smurfit Kappa on location in the plant in Italy. So I found that hugely insightful. So thank you for that, Luca. So next up, we have Nydia Lourenco. So Nydia is with NovaID, and Nydia is a senior researcher there. And in today's presentation, Nydia is going to share with you the work that they've been doing in NovaID on the development of green technologies for the isolation of PHA from microbial biomasses. So we can see your slides perfectly, Nydia. Everything is in order.
Can you hear me?
We can hear you now as well. That's perfect.
Okay, thank you.
Thank you.
So good morning. My name is Nydia Lourenco, as Elena said, and I'm a senior researcher at NovaID. And it's a pleasure to be here with you today and share our journey in the InGreen project. Can you see the slides?
Yeah, all good.
No, but I'm trying to move to the next slide, and it's not, can you see it?
I can see it. There might be arrows at the bottom of the slides, Nydia, just.
No. So just let me stop sharing and see if I can, okay. Let's see if we can see it now. Yeah. So today I'm going to talk to you about PHA recovery from mixed microbial cultures using green approaches. So I'm based at the Biochemical Engineering Group in the chemistry department of the Nova School of Science and Technology in Lisbon. And the experience of our team, well, we have over 30 years of experience in industrial and environmental biotechnology. We have participated in over 20 European projects during the last 5 years. And we also have some industrial contracts and intellectual property. And our facilities include lab and pilot scale bioreactors, and also different chemical and microbiological analytical equipment tools. And we also have advanced online processing monitoring tools, mainly near-infrared spectroscopy and Raman spectroscopy. So our expertise at NovaID include anaerobic digestion, PHA production, extraction and characterization, mathematical modeling, microbial polysaccharide production, and also process scale-up and online bioprocess monitoring. In the InGreen project, we have a task to develop an optimized eco-friendly methods for PHA extraction and purification from mixed microbial cultures. So as you saw in the previous presentation, Smurfit Kappa is producing in PHA-enriched biomass, and we will apply optimized PHA extraction protocols to this biomass to recover the PHA that is inside the cells. So at Smurfit Kappa, the biomass is produced, process has been optimized to enrich the microbial cells in PHA. So here we have an example of bacterial cells with PHA granules accumulated inside. And except for a few exceptions, like applications related to formulations for animal feed that you will also have the opportunity to see today, except for those applications where we can use directly the biomass with the PHA inside, we have to extract the PHA from the cells. And this downstream may represent up to 50% of the total PHA production costs. So this is a very important step in PHA production in order to allow application of PHA in many different products. Basically, there are two main downstream approaches. One is to solubilize the PHA granules, and the other one is to dissolve everything that is not PHA, so the non-PHA cell mass. So these are the major approaches that we can use to extract the PHA. The traditional approach involves organic solvent extraction to solubilize PHA granules, and then we need non-solvent precipitation step to recover the PHA. This traditional approach normally involves the use of chlorinated solvents like chloroform, and they have high PHA recovery yields, high product purity in the end, but most of these approaches are not eco-friendly, and most of them are not scalable or are very difficult to scale up. In fact, chloroform gives a very impressive results in terms of PHA extraction, but it's not allowed to use it at industrial scale. There are also organic solvents that are greener approaches. However, their cost is still very high, and also the equipment necessary to operate in order to perform this extraction is also expensive. So it's not a competitive solution at the moment for industrial scale PHA extraction. Our NovaID approach is based on chemical digestion to dissolve everything that is not PHA, and after that we'll do a PHA purification step. So we started to perform an extensive amount of experimental work at the lab scale, because as you can imagine, there are many different variables that we have to test in terms of the type and quantity of chemicals to use, the contact time between the biomass and the chemicals, and the temperatures of the reactions. So there are many different parameters that we can study and optimize in order to extract PHA. And only after optimizing a protocol at the lab scale, we moved into a pilot scale extraction. So in the next slide, you will see a video showing you how we start from the biomass produced at Smurfit Kappa and end up with a biodegradable polymer extracted and purified from that biomass. You will see in the video a step where we use chloroform, but I wanted to call your attention to the fact that this is only used for analytical purposes in order to quantify and or to qualify the final extracted product. And it's never used for extraction purposes at pilot or industrial scale. So I will show you the video that summarizes these steps for extraction, including lab scale tests and pilot scale trials. So thank you for your attention, and you can have my contacts if you have any doubts or questions. Thank you.
Thank you, Nadia, for that. Another fascinating presentation. And I think, sharing some really key areas around the development of eco approaches for the extraction of PHA, because when we do look at the production of these new bio-based products, we do need to take into account, I suppose, the full utilization of resources in the manufacturing process, from energy to the chemicals that are used. So that was really interesting, and thank you, Nydia. So if you have any questions for Nydia, we'll take them in the Q&A session, or you can pop them into the chat box as well, and we'll get to those in due course. So now we have a presentation from Irene Serrano. So we're now kind of moving on out of the PHA production area and into the more application focused presentations. So Irene is a research and development engineer. She's based at Tecnopackaging. And in this presentation, Irene is going to talk to us about the production of the biofilms from the PHA. And Irene is also going to share some insights from another BBI-JU project, which Tecnopackaging are part of, called the Uplift project. So Irene, your slides are up perfectly. So I think that's all in order. So I'll just hand over to Irene now. Thank you.
Okay. Hello, can you hear me well?
I can hear you perfectly, and we can see you, and your slides are up. You just need to do the slide share. That's it. Perfect.
There we go. So hello, everyone. My name is Irene Serrano, and I am a research and development engineer at Tecnopackaging. And today on this workshop, I will explain to you how the obtained PHA is transformed into biodegradable films for bag-in-box products, one of the many applications of the extracted PHA. But first, I will tell you a bit about Tecnopackaging. Tecnopackaging is a technology-based company located in Zaragoza, Spain. With a huge team, over 50 researchers and technicians, Tecnopackaging has given successful services to more than 80 customers since 2010. Our areas of expertise lay in the creation of advanced composites and blend. We assess the potential of new materials by analyzing their processability at laboratory scale, and we also can perform the upscaling, producing real functional prototypes. This is why we are involved in more than 14 European projects as developers of materials, technology, and first series of prototypes for companies as Nestlé, Eroski, Pascual, or Barilla, among others. Thanks to the confidence generated in our customers and due to our successful 12 years trajectory in collaborative projects within the European Union, we are able to invest more than 0.4 million euros per year in equipment, a number that is foreseen to grow in the upcoming period. Additionally, we belong to the Bio-based Industry Consortium, plus Ecolink, and we have been considered the Innovative SMA Award in Spain, where we were created. We are a proud member of the InGreen project, in which we are in charge of the production of the biodegradable films from PHA-recovered bacterial cells that are present in the paper mill wastewater biomass. The optimization and characterization of the material is also part of our work within the InGreen project, since they will be later transformed into bag-in-box prototypes to contain liquids, food, or cosmetic products. But let's stop here for a minute and have a more detailed view about the use of PHA. PHA is a bio-based and biodegradable polymer that has become very useful in huge sectors like medicine or the agricultural sector. Both of the properties of the PHA make it the ideal candidate to develop new solutions for packaging in order to reduce the dependence on the fossil-based or non-biodegradable current ones. The challenge is to manufacture bio-based films in a circular economy approach for the food, feed, cosmetics, and pharmaceutical sector. Packaging companies like the Smurfit Kappa group have shown interest in the development of new solutions, new alternatives to replace their current solutions. These bio-based and biodegradable films based on the extracted PHA will be used for the manufacturing of bag-in-box products, a type of packaging that is very popular in the sectors of food, beverage, and industrial liquids. But how do we go from the purified PHA to daily life, to bag-in-box products for daily life use? We use an extrusion compounding machine to blend the PHA with the necessary additives and a thermoplastic matrix that will enhance the properties of the extracted PHA and make it suitable for the final application. Then, the blend is tested in a film blowing equipment to assess its good processability. And after the production of the films, they are validated through mechanical, thermal, and chemical characterization to finally assess the suitability of the new developed material for commercial use. After the extraction of the PHA by our colleagues at NovaID from the organic waste, we are ready to develop the new material. This is achieved by an extrusion compounding process. Extrusion is a thermomechanical process in which a polymer is melted and mixed with one or more materials to change its properties or achieve new enhanced ones. The extracted PHA, a thermoplastic bio-based matrix, and the necessary additives are fed separately into a heated barrel that can have 1 or 2 screws continuously rotating inside. The barrel is also equipped with separate temperature controls that can be adjusted depending on the material that is being processed. The resulting high sharing forces between the screw and the material, plus the effect of temperature, leads to the melting of the polymer and makes possible the mechanical mixing of all the components. After being mixed, the resulting compound gets out of the extruder through a circular cross-section die in a shape of a continuous thread, and it passes through a water bath in order to be cooled down. At the end of the water bath, the thread is introduced into a pelletizer station and gets cut into small plastic pellets, ready to be converted afterwards into films. Once the compound is obtained, the next step is the film production. Film blow extrusion is a transformation technique in which a thermoplastic material, or compound in this case, is melted and then vertically extruded and inflated to create a thin wall bubble that will be later collapsed and wound up. This is the step in which the suitability of the material to become a film is assessed. The compound in pellet form is fed through a hopper into a heated barrel that has 1 screw rotating inside, and the material melts by action of heat and shear as it did in the previous step until reaching a viscous state. Then, after passing through the screw, the molten polymer is forced through a vertical ring-shaped die to form a thin walled tube. Air is injected from the center of it, making it expand and creating a bubble. This bubble gets cooled down by air, while it's pulled up by a series of rolls that are placed at the top of the film extrusion machine. Then these rolls collapse the bubble, giving it its final planar shape. And the newly formed film is taken down the top of the film extrusion machine, guided by a secondary series of bars, and it is wound up into rolls that will serve later for the production of the bag-in-box prototypes. Sorry, there we go. In parallel, we mechanically characterize the products obtained. We do this by performing film tensile test. Film tensile test is an experiment in which a pulling force is applied to a film specimen, measuring its response at each moment. It consists in a sample of standardized dimensions that is placed between 2 pneumatic grips with rubber ends in order to not let the film slip while the experiment is being performed. Then a pulling force is set by the machine, and the software of the tensile testing machine starts measuring the elongation of the sample in percentage versus the applied force measured in megapascals. The mechanical behavior of the material is represented afterwards by comparing these 2 values in what is called a stress-strain graph. Mechanical characterization is very important in order to check how the material will behave in case of straining, impact, puncture, etc. There is a huge spectrum of experiments that simulate the situations that the final product will be submitted to, but in a controlled environment. This experiment assessed the suitability of the newly developed material to become the final prototype. In this case, the prototype will be a bagging box to contain liquids, food, or cosmetic products. As a conclusion, I would like to say that in a world where the management of plastic packaging is one of the most discussed topics when speaking about sustainability, InGreen has provided valuable solutions. Sustainable tailor-made processes have been developed to transform residual streams into valuable resources for the production of biodegradable, eco-friendly packaging. And this is the end of my presentation. This will be the end of Techno's part. You can find me on the email written in the slide. And well, I give back the word to you, Helena. Thank you, everyone.
Thanks so much, Irene. That was really interesting, and it's just fascinating to see the development of the biofilms in situ in Tecnopackaging, and also the fantastic properties and results that you're getting. You can really see the progress that has been made through the InGreen project. So I think it's a really, really exciting time for the project, and just shows the good work that has been carried out. So thank you for that, Irene. So last and absolutely by no means least, we have Kim Wendy up next. So Kim is the Director of Operations at Avecom, and Kim is going to talk to us today about the use of PHA in the development of animal feed. Okay, so Kim, your slides are up, and just to press the slide share option.
Hello, good afternoon, everybody. I hope you can see my full screen.
Yeah, everything's perfect, and we can hear you perfectly, Kim. Thanks.
Okay. I will now go to the next slide, the title slide. So my name is Kim Wendy. I'm Operations Director of Avecom, and I will present to you how we can use the PHA-rich biomass in feed formulations. But first, I will tell you a little about Avecom. Avecom is an innovative R&D-driven SME company located in Belgium. Our company was founded in 1995 already and is a spin-out company of the Ghent University. The company was founded by Professor Willy Verstraete, a pioneer in environmental technology in Belgium and also abroad. We are specialized in the steering and optimizing of microbial processes. At the moment, we have a team of 10 full-time equivalents. On the pictures, you can see our founder, Willy Verstraete, but also our business developer, Stijn Boeren, at the bottom. In our facilities, we have several laboratories where we train bacteria for specific degradation or fermentation purposes. We have the possibilities to optimize our microbial processes from lab scale to bench scale, but also on pilot scale. Avecom developed the technology, we call it the PROMIC technology. PROMIC means the microbial protein technology, in which oxygen, carbon, and nutrients are upgraded to a high-quality end product. The carbon source in this case is originating from undervalued side streams from the food industry, namely starch-rich processed water. This technology uses an aerobic fermentation step with a specific microbial consortium. The downstream processing consists of a dewatering step, in this case centrifugation, and the drying step, in this case spray drying. Depending on the feedstock which is used in the fermentation process, the dry PROMIC product can be used as a protein source in food or feed applications. The PROMIC product is a non-GMO protein source and has general 70% of protein content. The amino acid composition, which is presented on the graph on the right side of the slide, is better than the composition of soy and approaches that one of high-quality fish meal. The product has an excellent digestibility. feed uptake, and also feed conversion. Previous shrimp tests showed that when shrimps were fed with ProMig as protein source, and when they were challenged with pathogenic bacteria named Vibrio, that the survival rate was a factor 2 better. Also, the weight gain factor was a factor 1.5 better. The product has shown to have immune stimulating properties. In the InGreen project, we combine this microbial protein source with the PHA-rich biomass that is produced from the process water of the paper mill factory of Smurfit Kappa. From literature, it's known that PHA can have a prebiotic effect on the intestines of animals, and therefore the addition of this PHA-rich biomass to the animal feed was investigated. We received from Smurfit Kappa and from Innoven the dried PHA-rich biomass and used this together with an external partner to formulate and characterize several feed prototypes, both for broilers and for shrimps. The optimal formulated feed prototypes were then provided to MTU. In vitro fermentation trials performed by MTU demonstrated that the prepared animal feed had a prebiotic effect. Based on these results, we have upscaled the production of the feed prototypes, and we produced larger amounts of the feed formulas to perform in vivo trials. The in vivo trials for the broilers are performed in collaboration with Lanupro, and the in vivo trials for shrimps will be done in collaboration with Imaqua. Just a small note, both companies are also spin-off companies of the Ghent University. We hope to present those results in a future communication. Thank you very much.
Thanks for that, Kim. That was really interesting to see, I suppose, the functionality really of some of the ingredients that you're working with and their opportunities around new product development. So that was really interesting. So thank you for that. So that's the end of the presentation session for today. So huge thanks to all of our speakers. Everybody was very punctual and kept on time, which was absolutely fantastic. So what we'll do now is we'll just go into our Q&A session. So just to our audience, please keep the questions coming into the chat box. We'll answer them as we go along. And I have a series of questions here. So I'm just going to kick off with some of the questions. So there's a question in here and it says that is PHA secreted to the broth as solubles or inside the microbial cells? So I think, Luca, this was one of the questions that came up while you were speaking, and I think Jesus, you might have some perspectives on this as well. So I'm happy for one of you to take that question, whoever would like to answer that one. So just again, I'll answer the question again. So is the PHA secreted to the broth as solubles, or does it stay within the microbial cells?
Morning, hello. I respond this question. Yeah, the PHA is the intracellular product. So when the PHA are synthesized, needed after digestion, for example, the biomass digestion for the extraction. Okay.
That's perfect, Jesus. Thank you. Okay. So we have another question. So this is from Luke. So are any of the paper mills involved using fungi to process their wastewater? So Luke, I think this one might be for you. So it's the use of fungi for processing wastewater. Is that something that you have looked at or something that you're aware of?
Okay. Specifically, we really don't know what kind of microorganisms we have in the reactors, because the point is that we use the mixed microbial culture, so we are not really interested in what kind of microorganisms specifically produce the PHA. We have an entire population made by different species that can produce PHA, but we didn't investigate the specific species that produce the PHA. It can be bacteria, but it can be fungi. We are not really interested in this information. I don't know if.
No, that's perfect, Luca. I think you've answered the question there. So thank you for that. So Irene, I think this might be a question for you, but our colleagues here as well might have some perspective. So feel free on coming in. So this is from Willem. And Willem's question is, so PHA is biodegradable and compostable, which is amazing, but it's also recyclable. Is keeping the material in the loop preferable to losing it to compost? And what are our thoughts around that?
Of course, the ultimate target will be to obtain a complete packing box made of PHA, but even though it is a material that is been widely used in other sectors, PHA is a huge family of polymers, and a bit more of investigation is needed in order to develop a whole biodegradable and compostable box. Of course, it would be ideal for it to be also recyclable. But I think that, well, it depends on the lifespan of the specific PHA that is being used. So I don't know if that answers the question, but yeah, the final target will be to have a complete recyclable, biodegradable, and compostable packing box. But it depends on the properties of the specific PHA that's being used.
No, I think that's really interesting, Irene, because, and I think you highlighted this, Jesus, as well in your presentation, is that that PHAs is a huge family of different types of molecules and polymers, and that bringing those together creates a huge variety of materials with different properties. But how you potentially might address that at the end of life, if you want to actually recycle and regenerate and reuse the PHA molecules, might present some other technical challenges that need to be overcome. So maybe that's another follow-on project from InGreen that maybe we can ask for some funding for, to look at that particular challenge. So I think that's really interesting. So Willem, thanks for your question on that. So I just have a question. So Nydia, you might be able to answer this for me. So the InGreen project is, you know, it's a large collaborative project. We have lots of companies that are distributed in different geographic locations across Europe that have to come together to collaborate as part of this project. And everybody kind of has a different position in the value chain or a different contribution to make. So I was wondering, could you tell us a little bit more about your experience, I suppose, with NovaID within this project, where you sit within the value chain, and what the experience has been like in managing to achieve this fantastic collaboration that we're talking about today?
Hey, Helena. Yes, we are in the middle of the value chain because Smurfit Kappa is optimizing a process to produce PHA-enriched biomass. And we are responsible for extracting the PHA from the biomass, and Tecnopackaging will use the extracted PHA in formulations to produce biodegradable films. So this project requires a close collaboration between the partners, and we have to discuss our results together very often. We have to share our ideas of optimization, because one change in the production process will imply changes in the extraction process, and will consequently imply changes in the production of the biofilms. So that's why we need to have meetings, frequent meetings, and to collaborate very closely with the partners in this value chain in order to attain a successful end product.
Very good. And then in terms of, I suppose, you know, after the project has ended and the kind of the scale up of the technologies and the processes? You know, what are the kind of key challenges that you might see, Nydia, around next steps, or do you have any comment to make on that?
In terms of, in particular, terms of extraction, you mean?
Yes, exactly. Yeah.
Well, it's already quite challenging because the behavior in the lab sometimes it's difficult to transpose to a pilot scale. And if you go to a larger scale, then the challenge is even bigger. In terms of the biggest challenge is to be in a chain where you have production process that is stable and consistent, so that the quality of the biomass that you need to process. And so that's the challenge for the successful extraction. And then to optimize at a larger scale the reagent consumption, the reaction times, in order and we also have to infer the quality of the extracted product, because sometimes we have a very good purity in the extracted product, but the extraction conditions might damage the final product in terms of molecular weight, for example, and then the final application might be compromised. So there are some steps that are challenging in scaling up this extraction to a larger scale, yes.
Yeah, so still some work to do, Nydia, to get
Yes, for sure.
I'm just wondering, you shared some really exciting product-related information in your presentation around functionality. So you shared the single-cell protein having really, really interesting immune stimulatory type activities. And then we have the PHA with the additionality of the prebiotic type activities. Just in terms of bringing these types of products to market, you know, are there challenges around communicating those functional benefits to potential end users and customers? And how does that relate back then to product price and affordability of these kind of new and innovative functional feeds?
Yes, of course, a further optimization of the applications is certainly required. We need to verify a few things on the in vivo trials, and hence also calculate production prices. Even though we work on recovered streams, it still has a cost to do a fermentation process, of course, and especially also downstream processing process. But we see, as Avecom, we see a big potential in sustainable feed ingredients with an added value. Immunostimulating compounds, less antibiotic use are all hot topics these kind of times. But also, we aim at stimulating the demand and also the public acceptance of recovered bioproducts. And I think it's something that we all have to do is to create economic value for all the business activities along these innovative value chains. Will customers pay higher prices just because it's because it's more sustainable? I think protein and also alternative ingredients, sustainable ingredients will come up the coming years, and hopefully we can also ask higher prices because of sustainability. But for now, it's we still have to see what the market will do with those kind of things.
Yeah, but look, we can see the trends already, Kim, around you know, security of supply of feed, looking at alternative protein sources. So I think the market is starting to shift significantly in the right direction. So I think it's an interesting time. Irene, Tecnopack is involved in quite a number of European funded projects and large-scale collaborations. And I think you're involved in a really interesting project called Uplift, I think it's called. Can you tell us a little bit about that and how it relates back to the InGreen project?
I think that you mean the Unlock project.
Unlock, absolutely. I keep calling it, it's another project I keep getting it mixed up with. Absolutely, yeah.
Don't worry, it's okay. Sometimes the name resembles each other a lot.
Yeah.
Well, Unlock is a very interesting project that is also funded by the Bio-Based Joint Industries Undertaking, and it proposes the revalorization of the further waste stream from the poultry sector in order to design new economically and sustainable value chains. This is achieved by the extraction of the keratin that is present in the feathers from chickens or poultry sector, which is approximately 90%. I mean, the feathers have approximately 90% of keratin. And this keratin is used for the creation of new bio-based and biodegradable materials and products for the agricultural sector. In relation to this, PHA is also a biodegradable polymer that we've been talking about, and that is widely used in the agricultural field due to its properties. It does not harm the soil, and every biomass that is harvested with PHA on it can be directly composted as organic residue. So PHA is highly interesting for the end products of the Unlock project, which are seed trays, hydroponic foams, geotextiles, or mulch films. These are products that help the growth process of the plants and crops, and once their lifespan has finished, they can be returned to the soil, and the keratin that is present on it serves as a release agent of organic nitrogen, well, implementing a circular bioeconomy. It's very similar as what InGreen does.
That's really interesting, Irene. Thanks for sharing that. It's just amazing the variety of applications. PHA really seems to be one of these wonder products that I think we're going to hear a lot more about in the future. So that's actually all we have time for in today's event. A huge thanks to all of our speakers for such fascinating and interesting presentations. You've really brought us on a journey from side stream right up to the market, and it's been hugely enlightening, and I think has really showcased the fantastic work that the InGreen project has been doing. I'd like to thank Eve Savage as well for all of her fantastic work on coordinating and getting today's event together. So thank you to Eve, and I'd like to thank everybody as well for taking the time to attend today's session. It will be available as a recording, and there were a few questions that we haven't answered, so we'll make sure to get the answers of those questions out to you. And we will be hosting another event that will be showcasing the third value chain in this project in a couple of weeks. So please keep an eye out for the dissemination and promotion of that event. And thank you to all of you for attending today's session.